Welcome, everyone. Happy to welcome today Ariana Marquez Gonzalez from Jason Stein's lab in North Carolina. Both myself and Ariana are here in Bordeaux at the end of the Cajal course. And I see several of the students and instructors have also jumped on. So, welcome to everybody that's actually local here in France and to everybody else that has joined. Jason Stein and his lab have been for years studying many different ways of studying gene by environment interactions in vitro. And Ariana today is going to share, I think, the most recent iteration of some of those investigations. Thanks so much for joining us, Ariana, and take it away.
Great, thank you, Eden. Hello, everyone. Thank you, Carlo, for the opportunity to share my work with this community. As Carlos said, I'm in a Stein Lab. I'm a PhD student in the neuroscience curriculum at UNC Chapel Hill and working under the mentorship and guidance of Dr. Jason Stein. And today I'd like to talk about my project studying gene bioenvironment interactions using human cortical organoids in the context of autism spectrum disorder. And this is our work in progress. So unfortunately, we don't have much results yet, but hopefully I can convince you about the approach and why this project is important for the field. Oh, and please interrupt me at any point or send your questions. I'm happy to answer the questions. Um, okay. And then to start, let me define what I mean by gene by environment interaction, or GBI for short. And a GBI occurs when the effect of an environmental exposure on a trait depends on an individual's genotype. And for example, here we have a locus where, okay, that's a right slide. Here we have a locus where three individuals carry different genotypes, AA, GA, and GG. And at baseline, they look identical. And it's only in the presence of a particular environment, which you already saw by mistake, that differences in phenotype emerge. And like since you already know, we're presenting from France, so I won't waste the chance to use a croissant as my exposure. And in this example, the G allele confers susceptibility to the croissant. So individuals that are carrying two copies of the G allele show the strongest response. And the trait can be anything like fluency in French, for example, or more relevant, like today's talk, the development of disorder. So there are many disorders that have both genetic and an environmental component. In autism specifically, the genetic architecture is complex, and this plot captures that. So in the x-axis, we have allele frequency, how often a variant is seen in the population. And on the y-axis, we have the effect size, so how strongly that variant influences disease risk. And what we see is an inverse relationship. Rare variants tend to have large effect, while common variants have small ones. So although the novel and rare inherited variants are essential for understanding the molecular mechanisms of autism spectrum disorders, most of the heritability is actually explained by common variation. And this is where genome-wide association studies, or GWAS, have been essential. They let us pinpoint the common genetic variants associated with ASD. And in case you're not familiar with this Manhattan plot, each dot is a genetic variant. The x-axis shows genomic position by chromosome, and then the y-axis shows a statistical significance. When a dot is above this line, which is the threshold, that variance is considered significantly associated with the trait, or like with ASD in this case. But the question becomes, what happens when we try to interpret what these variants actually do? So often we see something like this. Even though one of these variants is associated with ASD, there's no detectable difference in gene expression between the two alleles. So like, how do we make biological sense of these hits? And the key is to remember that gene regulation is highly dynamic. Expression can vary by cell type, by developmental stage, environmental stimulus. So the reason we don't see a difference in expression at baseline might simply be that we are missing the right context. And once we have the right context, now we can interpret like what these variants are doing. And we can study different brain cell types, different neurodevelopmental windows. And like, this is where the environmental becomes especially important because environmental context may help explain part of the so-called missing heritability in complex disorders like autism. But with so many exposures, possible exposures, where do you even begin? And like fortunately, decades of epidemiological work have already given us strong candidates, and studies have identified like a range of environmental risk factors associated with autism, including birth complications, prenatal exposures like air pollutions, pesticides, plasticizers, or like even medical conditions like diabetes, fever, infections. And among all of these, the most replicated single compound environmental risk factor is valproic acid, which is an anti-epileptic drug. So I'll be focusing on this today. And valproic acid was first described as an anticonvulsant in 1967, 1967, and it later became widely used and is now FDA approved for epilepsy and bipolar disorder. The link to autism was first reported in 1994, but it wasn't until around 2011 that became evident. And this was because larger studies began to define a clear prevalence. For example, in a cohort of 580 prenatally exposed children, ASD prevalence was about 4% compared to roughly 1% in the general population. And mechanistically, BPA increases GABAergic activity and acts as a histone deacetylase inhibitor, altering chromatin accessibility and gene expression. However, like prenatal exposure to valproic acid is associated with teratogenic effects, neural tube defects, and a significantly increased risk for autism. So, given these risks, the FDA has placed valproic acid in pregnancy category X for migraine treatment, which means that the benefits do not justify the risk of malformations, and will remain in category D for treatment of seizures and bipolar disorder. So pregnancy category D indicates that there is positive evidence of human fetal risk, but the benefits from using pregnant women may be acceptable despite the risks. So if the drug is needed in life-threatening situation or for a serious disease for which like safer drugs cannot be used or are ineffective. And so we know when pregnant women take valproic acid, their children have a higher risk of developing ASD. But, and this is the key observation, not all exposed individuals develop autism. So that variation in outcome is what motivates our central research question: is underlying genetic variation responsible for driving these differences in susceptibility? To begin answering these, I want to talk about recent work from our lab led by two previous grad students, Ran Lee and Jordan Ballon, and our bioinformatician expert, Nana Matova. In that study, they exposed the primary human neuroprogenitor cells from 80 individuals to valproic acid and lithium or vehicle for 48 hours. And then they measure chromatin accessibility with ataxic and gene expression with bulk RNA-seq. What they wanted to study are cases like this, where in the vehicle exposure, there is not really a change in chromatin accessibility or gene expression, but in the stimulation, there is a change depending on the genotype. So something like what I showed you before with the croissant example. And here's what they found. So both context-dependent EQTLs for the expression and CAQTLs for the chromatin accessibility, meaning that genetic variants that affected chromatin accessibility or gene expression only upon drug treatment. And so here that we have is on top, we have the valproic acid. And then this is Miami plot where you have the response QTLs for valproic acid on top, and then the vehicle response QTLs on bottom. And then we have the same for lithium and on the bottom. So next, I wanted to integrate these context-dependent QTL with GWAS for relevant traits. So they run TWAS in each condition, valproic acid and vehicle, and look for genes that could mediate the impact on cognition, because prenatal exposure to valproic acid is also associated to intellectual disability. And what you can see here is that those that are on purple are those genes that are associated with either intelligence or education attainment, and they are highly expressed in the, not highly expressed, sorry, differently expressed in the valproic acid condition. And when you look at these genes, you see that they are enriched in pathways associated to folate. Okay. So the other thing that they did is that they look at these EQTLs and they did something that it's called colocalization with GWAS. So here we have that the genetic variant that is susceptible to valproic acid, it also colocalizes with a genetic variant associated with educational attainment. And this particular case is MTHFR1, which is a gene associated in folate metabolism. So like, this was very interesting for the lab, since like folic acid supplementation along with related vitamins was actually shown over 30 years ago to rescue VPA-induced teratogenicity in mice, and in humans, folic acid supplementation during pregnancy has been associated with a reduction in autistic traits in their children. So altogether, this GBA dish led us to a targetable treatment pathway that has already been validated in both mouse and human. So, okay, that is a study already published, and I want to talk to you about my project, which is built upon like this previous study, and we want to expand these efforts in several ways. So the first one is that now we are using a different cohort, and this will increase our analysis because now we'll be able to do a meta-analysis. So we are using 115 iPSC lines from the infant brain imaging study, and I will talk a little more about that later. Now we are changing our model. Before it was human neuronal progenitor cells. Now we're doing iPSC-derived cortical organoids, and because we will do single-cell RNA-seq, this will allow us to investigate cell type specific responses. For exposures, we are doing 2 doses. And we are also doing 2 terms, repeatedly exposure for long-term for 7 weeks and short-term for 48 hours. And on top of that, because our cohort, we have clinical data from this cohort, we will be able to correlate VPA-induced responses with clinical data. Okay, so first I want to talk a little bit about like what IVIS is, because we are using these cell lines from this fantastic resource. And some of you may already be familiar with IVIS, but for those who aren't, IVIS is a longitudinal initiative designed to identify brain changes that occur during development before an autism diagnosis is typically possible, which is usually around age 2 or later. To do this, IVIS recruits families that already have an older child with autism and a newborn child, because the newborn shares similar genetics and similar environment with their autistic sibling. they are considered to be at high likelihood to develop autism. The study then tracks the new baby's development with MRI scans, cognitive scores, and when ultimately an ASD diagnosis. Because we collect blood samples from these individuals, we can reprogram them back in time to have these iPSCs. And what makes this cohort uniquely valuable is not just the genetic variability across a large sample, but the opportunity to correlate our in vitro findings with rich clinical data from the same individuals. Across our iPSC cohort, we therefore have 3 family categories. Those individuals that develop autism, those that didn't develop autism, and as negative control, individuals from family with no history of autism. And because we got to compensate like the relatively low number of ASD positive cases, we also included some samples from the older sibling, affected sibling. And just to be clear, we won't have information, MRI information from the sibling, but we have the unique opportunity to interrogate shared versus divergent responses. So how do we leverage natural genetic variation across these many individuals? And this is because each person carries a unique combination of variants, and that's what's shown here. So across persons, person 1 through person N, we take a blood sample from each donor and reprogram those cells back into induced pluripotent stem cells, and then differentiate them into cortical organoids. Because these organoids retain each donor's genetic background, they let us model gene bioenvironment interactions in a control human neurodevelopmental context, which is exactly what we need to address our question. Now, in terms of the experimental design, once we generate cortical organoids from these lines, we expose them to one of 2 paradigms. For the first one, we expose them to vehicle in the 2 concentrations of valproic acid from day 35 of differentiation to day 83 of differentiation. And this is a repeated exposure that mimics closer what happens in vivo. And then every week we acquire right field imaging from these organoids. Then for a short-term exposure, we expose the organoids at day 81, just 48 hours before collection. And because we have the right field imaging, we can look at the cross-sectional area of these organoids and see how they respond upon the 2 doses of valproic acid. Then, at day 83, we dissociate the organoids and we fix them for single-cell RNA-seq. And then eventually we will be able to identify valproic acid response EQTLs, where again we have the vehicle condition with similar levels of gene expression. And then upon valproic acid, we see differences in gene expression depending on the genotype. Okay, so after a lot of cell culture and a fantastic team, we ended up generating organoids from 131 unique cell lines. These include other cell lines outside the IV's cohort. And in this experiment, we have a subset of donor replicates. This means that from 10 individuals, we had 3 iPSC clones, and we generated organoids from each of the clones. And then we also have clone replicates, where the same iPSC line from the same individual, and from that we generated organoids 2 different times. So in total, we have around 160 differentiations, including the replicates. And to our knowledge, this is the largest cortical organoid data set of its kind, and it gives us the power to look at response CQTLs across two doses and two exposure durations. So for the library prep, we are using this split-seq method from Parse Biosciences. Briefly, organoids are dissociated and the cells are fixed. And then through several rounds, we add different barcodes. And what happens is at the end is that each cell will have a unique combination of barcodes. The resulting cells are pulled into sublibraries, then their cDNA is isolated and prepared for sequencing. The advantage of this approach is that we can multiplex enormous numbers of cells across many samples in a single run, which is essential at the scale we are working right now for this project. On the sequencing side, we made a choice to compare two platforms. Before, we were using Illumina to sequence our data. However, because we knew about this new platform called Ultima, and the prices are very accessible, we decided to compare these two platforms. And this was done by Nana. And what we can see here is that we sequence the same sublibrary with the two platforms, and we have a high correlation in the number of counts and Gs across the two platforms. Then for this sublibrary, we had organoids on day 84 and day 14, and Nana looked at differentially expressed genes, and we see very, very similar results in these volcanoplots. And this volcanoplot is from the Ultima, and this is from Illumina. Then lastly, here we see the log fold change of these genes. And again, we see a higher correlation across the two platforms. This agreement also holds at the level of cell type assignment, or at least like cells assigned to each cluster, since we haven't annotated these cells yet. But here we have the clusters from Ultima and then the clusters from Illumina. And most of the cells are assigned to the same clusters with slight differences. So right now we just got the data back like maybe a month ago. And because everyone loves to look at UMAPs, here is our UMAP for one sublibrary. But we will, at the end, we will have 10 million cells at a sequencing depth of 35,000 reads per cell. And right now I'm just talking about valproic acid, but we are also doing other conditions that will be included in this data set. And when we look at the expression of canonical cortical markers, or organoids capture the full diversity of cell types you will expect during human cortical neurodevelopment. We see neuronal progenitors, intermediate progenitors, outer radial glia, markers for upper layer neurons, and markers for deep layer neurons. And as I said, we are analyzing the data, so I don't have a lot to show, but here we did a preliminary analysis with only 4 donors, and we look at the differentially expressed genes after short-term treatment with valproic acid with the highest concentration and long-term treatment. And what we see in the gene ontology terms are terms related to synaptic signaling and oxygenesis, and also genes that are enriched for synaptic vesicle pathways. The other thing we were able to do is compare these differentially expressed genes with the previous data from the study that I mentioned, and we see a high correlation of upregulated and downregulated genes from the bulk RNA-seq data from the human neural progenitors. And beyond transcriptomics, valproic acid also has a clear morphological effect. So here we have our organoids treated with either condition of valproic acid. And we see that those dependent response where those organoids treated with valproic acid have a smaller cross-sectional area in after long-term exposure. But here is where it gets interesting, and we are very excited about this. If you look at individual donors, you will see something like this, where there are some donors that seem resilient to valproic acid, and they don't have a huge change or difference in area at any of the doses compared to vehicle. But then you see other donors that look like they are more VPA susceptible. And right now, we are still blinded to this analysis, so we don't know the genotype of the donors, but we will see if there are genetic variants that are driving this effect. So for future directions, first we'll do the EQTL mapping in our single cell data to identify BPA-dependent genetic variants. Second, we'll test whether those same variants colocalize with ASD and brain-related GWAS signals. And lastly, we will look at the correlation with the clinical data to see if VPA-induced responses correlate with IV's cognitive scores or MRE data. So in summary for this section, building on prior evidence, the GVI effects can be recapitulated in vitro, but we've been calling the GVI in a dish. Our approach provides a controlled and reproducible system to dissect how genetic background modulates response to environmental insults. We have generated a single cell resolution dataset to investigate valproic acid susceptibility. VPA exposure induces global transcriptomic and morphological alterations, and we observe cell line dependent response to valproic acid, suggesting genetic modulation of effect size. But this framework is not specific to valproic acid and can be applied to different environmental exposures. So right now I talk about valproic acid, but we also are trying to model lead to represent the heavy metal exposures, PM 2.5, to represent air pollution. PM 2.5 stands for particle matter 2.5. And PFOA, which is the forever chemical. This is known as that because it can be found in non-stick cookware or waterproof products, and also in water, in drinking water. And we can also model biological responses such as interferon gamma to mimic infections during pregnancy, or hydrocortisone to mimic stress. So here is just another small pilot that we did where we exposed our 4 donors to interferon gamma. And what we see is that there are upregulated genes associated to immune response. So it's what we expect. And then for PM 2.5, again, to mimic model air pollution, we look at metal-related genes, just because PM 2.5 is a mix of different stuff, different things, and one of them is metals. So we have this existing data set where we have organoids exposed with vehicle, valproic acid, lead, PFOA, and hydrocortisone. And then we are starting this new exposure panel where we are including PM 2.5 and interferon gamma and some 4 other compounds that we haven't determined yet. And the impact of this project is to establish a framework for the study of gene bioenvironment interactions. We want to understand the molecular mechanisms in autism, And ultimately, we want to inform personalized strategies. So in summary, the talk overall, iPSC-derived cortical organoids maintain the donor's inherent genetic background, serving as a powerful model to study the interaction between environmental stressors and common genetic variation. This framework is scalable and can be extended beyond VPA to study a wide range of environmental exposures relevant to neurodevelopment. And altogether, cortical organoids offer a unique platform to model human neurodevelopment during pregnancy and to study how diverse environmental stressors interact with genetic variation to influence ASD risk. With that, I'd like to close by thanking the people who made this work possible, Jason, Nana, and the people working closely with me in the cell culture, Esther, Alvaro, and Rubo. Thank you to my thesis committee, to our collaborators, Ramona Optics, which they gave us the opportunity to image all these organoids, the UNC stem cell core who generated the iPSC lines, and of course the IVs family and our funding sources. Thank you.
Thanks, Ariana. And now please jump in with any questions.
Maybe I can stop sharing.
Sure. So I have a general one maybe to kick us off. You mentioned the number of lines, 115, which is, I think, for all of us sounds very large and daunting. I know you mentioned a little, I don't know if I missed some of the details. Could you give us an idea of the practicalities of growing and treating and sequencing 115 human lines? I believe individually, correct? There's no, yeah, we'll go for it. Why don't you tell us?
Yes. No, of course. Yeah, that is correct. These are individually, and it was a lot of work, and it wouldn't be possible without this amazing team and without Jason's guidance. But what we did first is that we differentiated these cell lines in different rounds, and every round had between like 10 to 15 individuals. Then we make sure that we are blinded to the diagnosis, but we work with the IV's clinical team to get even distribution in diagnostic groups in every round. And we had to be careful with to reduce like technical like batch effects. And so we just write down everything, like who took care of each step. We make sure that we had the same lot number for most of the reagents, and if not, we will take notes for that. And it was fun. Then of course for the library prep, those were long days, but like really something that helped us is that every fixation we culture the organoids individually, but at the moment of fixation we pull the organoids together, and then we are going to use the genotype information to the convolute and assigned donor identity.
I think I'm not a wet lab person, but I'm sure that sounds crazy and daunting and quite an accomplishment for those that spend time in the wet lab. Pau, please jump in.
I mean, I just see a question in the chat, so maybe let's do that first. Do you think?
Okay, sure. I'll read that out. So the phenotype of the VPA susceptible organoids is intriguing, particularly because they appear smaller. However, how do you exclude the possibility that this observation reflects normal organoid variability rather than a true phenotype?
Okay, yeah, that's a great question. So first of all, it's been reported microcephaly organ that brain size changes upon valproic acid exposure. And so we do expect this phenotype, the microcephaly. But yeah, the other thing is that you're right, like there is some variability happening when culturing organoids, especially when you're culturing these many cell lines. Thankfully, we have for every donor, we also have bright field images for like 7 additional unexposed organoids, which eventually will become the short-term like exposed organoids. So we are gonna look at those, and we expect that those or unexposed organoids are more similar to the vehicle rather than the valproic acid. So that's our like way to isolate this variability or look at this variability in the lines.
Great. Okay, I have three very quick questions. The first one is obviously the organoids, you can differentiate it for whatever number of days. I don't know what's the limitation there. And you mentioned very well that the interpretation of the GWAS hits depends on the cell type, developmental stage, and stimulation. So I was wondering, how do you pick up that time? Because obviously, when the organoids can recapitulate autism disorder onset or whatever that causes the disease later in life, how do you choose that day?
Yeah, of course. So for the start, yeah, there are many like different exposures that you can do, and it can start different time points. You can expose the organoids, then remove the exposure. There are definitely different designs. For us, we started at day 35 because at this stage we see both progenitors and neurons. So we wanted to capture the exposure in both. The other thing that it's very tricky is that if you expose the organoids early in development in culture, then this exposure, you don't know if it will affect the differentiation. So at the end, you might end up with cells that are not important for your experiment or for your question. And yeah, I think that day 83, it was chosen based on previous work in the lab. It will be great if you can culture the organoids longer and then have more astrocytes, but it's scary with this number of cell lines.
Yeah, actually, the second question was related to this massive size of the data set. And one of the things that happens with an egg is that you like somehow design an experiment and you tolerate a range of the number of cells you can see for each donor in this case. So what is the lowest number of cells that you have set to see? Okay, with less than these cells, we cannot see the phenotype we would like to see. Or is there a number or it doesn't?
for the single RNA-seq
Yeah.
data, I'm sorry, I'm not sure I understand what is a minimal
So
number of
you have
cells.
said that you expect around 35, you have designed for 35,000 reads per cell, but then obviously there is a number of cells per donor that you aim to see in the, right? So,
Yes. Yeah. We are aiming for 5,000 cells
okay.
per donor.
Wow, that's a lot. Okay, good. And the third question was linked to the final experiment. What are the kind of phenotypes you want to see in the single cell data, or we expect? Like, it's like changes of cell types, or kind of now that we have kind of here, kind of patterns change in the transcriptional signatures, or a bit of both, or
Yes, I think it will be the thing that we are gonna look at is cell type proportions. See if we have like as cell type proportion changes upon valproic acid exposure. We are also gonna look at the gene expression, and definitely because with the progenitors we found these changes in genes related to folate. We can see if we can replicate that.
thanks.
Great, thank you.
I had sort of a similar question, but first I just want to say fantastic talk. This was lovely. And as someone who does iPSC cell culture, I am incredibly impressed. with the amount, like the scale and like all the factors that you're including in these studies. I think it's going to be a fantastic data set. And I'm really curious if you have thoughts on almost like expected cell types that you think will be most affected by VPA, since you're getting this sort of heterogeneous mix in the organoids. Are you expecting there to be particular cell types most vulnerable, some sort of a mix, or maybe different responses in different cells? And I'm curious too about, I love the context dependent aspect of this and the idea of actually being able to get sort of context QTLs with the treatment. But I'm curious if you think you have the power to also do sort of a two-way intersection of both cell type specific QTLs and context QTLs like combined in some way to get even higher resolution with this data set.
Yeah, thank you so much. Okay, so the first question, I think we do expect, well, I expect that progenitors will be a little more vulnerable than neurons. I'm interested in looking at neurons and see how, like, if there is this change in genes associated to neuronal activity, because we see like these changes in synaptic vesicle. Also, like valproic acid promotes neurogenesis. So like that's the population that I think it will be most affected. And then for the other one, we also thought a lot about these. We do think that we will have enough power to detect these cell type specific context dependent QTLs. But at least we are very confident that we can divide our population in neurons and progenitors. But we will look at if we can go further and look at very like more specific sub cell types. That will be amazing.
Gotcha. Thank you.
Thank you.
So I see here in the chat, and Ron, jump in if you'd like to. Ron Mackay has a question that relates to the previous VPA-induced organoid size phenotype. He says, this is an interesting question. To add to the discussion, the Vallone et al. Paper shows a dose response for cells in S phase with increasing VPA exposure. And Ron, if you feel like unmute, jump right in. So yeah, obviously those are the size in the. Oh, jump in, Ron. Got it?
No, I was just going to say, it's very interesting. Thanks so much.
Having a little trouble hearing you, Ron. Is anybody else having trouble hearing him? Or is that just me?
I can hear, yes.
Okay, go for it, Ron.
Okay. In the previous paper, it was a very short term. The cells that you were treated for 48 hours after a kind of neurosphere expansion. And here, we're looking at organoids which have been cultured for many days, 80 days, 83 days or something. And what I was wondering about is that in the previous paper, which has a lot of work in it, so I haven't completely grasped it, but it seems like the evidence for the folate metabolism target or sensitivity is derived from that short-term experiment, right? Where it's clear that VPA is having an effect on proliferating cells from the S phase result. But what am I trying to say here? That I think one of the reasons we're so intimidated by this question is that you have this whole period of differentiation of neurons during pregnancy, which is where you might expect susceptibility to occur at many different stages, and many different teratogens or toxins in the environment are apparently contributing to autism risk. And yet here, even with a short-term treatment, you make real significant progress. And now with this longer-term study, it makes me think of asking whether or not you think that the folate metabolism is a final common pathway for all these diverse potential toxins Could it be much simpler than we think?
Yes, no, yeah, that's an excellent point, and we were very excited about this because when it comes to thinking, okay, how can we solve this? Like we already know, like this is causing this. Now we know like the genetic variants that are susceptible, but how can we uh, target these with a medication, especially because during pregnancy you don't want to take medicine and like external medicine because it might affect the baby. But now if you have this folic acid, this is already recommended for pregnancy, and now like you have these genetic variants, and in you look at these genetic variants in the pregnant women, and now maybe like you, because you are more susceptible, maybe we can increase the doses of folic acid, which is already done for certain conditions like um, certain medications. So it will be amazing and very impressive if we can find these shared pathways with, especially with the other stressors that we are looking at.
Yeah, it's provocative. Thank you.
I might jump in on that with just a general comment about a convergent pathway across multiple kind of neurodevelopmental risks, and might there be one common theme between them all. And I actually participated in that S phase experiment you were talking about, Ron. And we were really surprised at how just strong of a drop in S phase that was. We thought maybe VPA was killing cells, there was some toxicity effect, and it was definitely not associated with any sort of toxicity. It was specifically about inducing neuronal differentiation. And so the plots that Ariana showed, where you have hundreds of genes going up and down in response to VPA. This wasn't necessarily that neuroprogenitors were changing their expression so much. It's that they were differentiating into neurons. And so as part of that transition, gene expression massively changes. So VPA, inducing a kind of a cell fate decision to become postmitotic and turn into a neuron, that differentiation process and that differentiation timing may be the common thread across all of these, is that neuroprogenitors being in a neuroprogenitor state and deciding to become postmitotic, it's not a molecular pathway per se, but definitely a shared pathway that could be common across lots of different environmental conditions or environmental challenges.
And speaking of convergence, I think given the epigenetic mechanistic knowledge we have about VPA, there have been many studies in other chromatin handlers that have shown precocious neurogenesis. So this again could be a convergent problem. You don't, waiting and being slow is one of the things that is key to making a human brain, and speeding things up can really be a problem, and potentially, as everybody's sort of hinting at here, maybe a common problem across different risks and impacts here. Very cool.
Going in the published paper, you compare lithium and valproic acid, and it's very interesting looking at the top genes that are affected by these two treatments, and it made me wonder whether this effect on the transition out of a proliferative state, that we need to think about it extremely carefully. What are the cells actually doing then? Because in the lithium treatment, one of the genes that's turned down is OTX2, but lithium is reported in that paper to increase proliferation, and so the cell that's proliferating there is at the same time turning off OTX2, which I associate with an early progenitor stage.
Yeah, that seems backwards, right?
It seems like the intermediate cells, what are those intermediate states that are proliferating or differentiating, and what decisions are they making as they go through that transition? Maybe I'm just showing my bias towards being so interested in this event, but it seems very, very cool what you're reporting. Thank you, anyway.
Thank you. Yeah, and talking about opposite effects, We were also looking at, this is something that we need to investigate further, but we were looking at like neuronal activity upon valproic acid exposure. And we will look, we saw the reduced neuronal activity, which is consistent with the anticonvulsive properties of valproic acid. But then when we were looking at the literature, we saw that there was one paper that reported actually the opposite, the hyperexcitability. So now we are wondering, like, okay, so one thing is looking at the exposure, but then you have to look at different developmental windows and maybe different concentrations. So there's definitely a lot of there's room for like more exposures, more conditions, more at different like time points.
Your single cell data should tell you whether there's an effect on the relative numbers of excitatory and inhibitory neurons, which would be extremely interesting. Yeah,
Yeah, definitely.
Sort of maybe playing on Ron's thoughts about early states. I was wondering, I think you said, looking at the times you're looking at, the sequencing and most of your phenotyping, the organoids is, what is that, almost at 90 days somewhere, right? 83 or close to that, 85. In this big cohort, have you thought of, or is there a hope or a plan to get cash and support to do some measurements earlier in the both pluripotency and early neuroectoderm emergence from pluripotency in these same lines?
Yeah, that will be interesting to look at. There are definitely studies where VPA it's added to younger cultures and they become.
Well, I was actually thinking of before you even challenge them to get a sort of a deeper characterization of molecular environments that may set up susceptibility when you later hit them. So untreated pluripotency or untreated early neuroectodermal states.
Yeah, that will be very interesting. I'm just trying to think a little bit more on how that will work. I guess like how could we determine susceptibility even
So
like before?
you would have the information about which of your 115 lines are hit hardest and in what way with your data you just described. And if there is a state, an early state, whether it's pluripotency or early neuroectodermal state, that travels with that susceptibility, that would be super interesting.
That's specifically very interesting for those families that you have shown that some of them have autism, others do not develop autism despite the risk, and others do not develop it at all. I think in the risk wall, that would be a very interesting thing to do at IPS states, because obviously there is something that explains the susceptibility. There has to be.
Yeah, that will be amazing. And actually, I think we can look into that because we have bulk RNA sequencing of the iPSCs before the induction, and then Esther and Ruel, yes, and then Esther and Ruel are doing like, no, definitely, we need to talk more about this.
Wonderful, that's absolutely.
Yeah, they will also have a single cell RNA-seq data of this cohort at day 14 of differentiation. So that's and these are the same cells because we differentiated everything together, and then I took the cells for the GBIX projects at day 35.
That's great.
Can I ask a quick question?
Yeah, go for it, Paul.
No, I remember at the beginning you talked about the different genetic architectures of autism, and I was wondering, the lines that you have generated, do all of them come from a same architecture more or less, or is, because I guess that also introduces heterogeneity in the things that you are measuring with environment.
Yes, this is hepatic ASD, and my understanding is that we don't have any like rare variants.
Thanks.
You're taking on the hardest problem. Any other questions? This has been fun to discuss, Ariana.
We can continue.
And we can continue our dinner.
That's right.
No, thank you so much for the opportunity. Those were great questions, and thank you for joining. This has been amazing.
Okay, guys. Cheers, and yeah, we'll see you at dinner. And thanks for coming, everyone. And we'll let you know. Maybe we'll bug you again, Ariana, when you get further down the road with your results. We'd love to see some more. So keep us
I would
informed.
love to share it. Thank you. Thank you, everyone.
Great. Bye, everybody.